In this video we are going to talk about the neurons and how they, with the resting gradient and action potentials, doing their job. Neurons are the cells, the main assignment of which to transfer the information around the body. The information is sent with electrical signals, action potentials. A simple model of the neurons consists of four main parts, which allow the neurons to be fantastic at this job. The dendrite receives signals from the neighboring neurons, like a radio antenna or radio receiver. The axon is the long output process of the neuron, which transmits signals and action potential over a distance like telephone wires. The axon terminal, which is endpoint of an axon, transmits signals to another neuron dendrites or tissues, like a radio transmitter. Myelin sheath is an insulating layer and actually speeds up signal transmission along the axon. Let me figure out how is the neuron functioning. Let us think about the three potentials. Resting potential when neuron is in the rest, although it's not really the rest. Graded potential with based on the stimulus that neuron will get. And then we may have or not action potential that can lead to synaptic activity on the next neuron. We are going to start with the resting membrane potential. If we look on the cross-section of neuron, it maintains a fairly consistent negative concentration gradient between -40 to -80 mV. In a typical human neuron, it is -70 mV. On the action potential diagram, on the either side of the action potential, the neuron is at the rest. Let's zoom on the surface of neuron to see resting potential in details. In order to remember the ions on either side of a membrane, it is helpful to use a mnemonic salty potato. Most people remember that potato is rich with potassium, and neuron has a high percentage of the potassium inside. It does not mean that it does not have potassium outside, just percentage of potassium inside is extremely high. Proteins give also negative charge ions inside of neuron. Since it is salty potato, we have sodium and chloride ions on the outside of membrane. We still have them inside, but in significant low percentage. But if we look carefully, there is still no potential. We do not see a separation in charges. In order to figure out it, let's remove everything to focus on the potassium, because the potassium and membrane permeability to the potassium establishes the membrane potential. The neuron membrane is super permeable to potassium ions. Lots of potassium leaks out of the neuron through holes in the cell wall, potassium leakage channels. Since there is a chemical gradient from higher concentration inside to low concentration outside membrane, that encourages the potassium to move in the outside direction. What happens when the potassium moves out? We see the separation in charge, since it is positive charge outside and relative negative charge inside. We also see the arrival in new electrical gradient. Since we have all positive charges outside, they can push away each other and move towards the negative charge. So, as more potassium leaves, then electrical charge is increasing. Eventually, we have movement of the potassium outside and inside. For all that, we got the electrochemical gradient outside. Why don't all potassium flow out? It is because the leaked channel doesn't allow much of the potassium to flow out. Now, if we look on the fat simulation, we see the passing of the potassium inside and outside of the cell. Actually, we also have another leak channel. It is sodium leakage channel, and sodium ions slowly leak into the neuron through it. So, we have an electrochemical gradient of sodium. Remember, there is more sodium on the outside that's moved in. Therefore, we have two sodium and potassium electrochemical gradients that are set up. We cannot see the chloride and protein moving. That's because we don't have channels for them. If we would continue this way, eventually we would lose all potential, since all the sodium would come in. So we have sodium-potassium pump where it moves the sodium out. Actually 3 sodium ions for every 2 potassium ions. Therefore, the sodium-potassium pump is basically keeping up with sodium flowing in and potassium flowing out. And so that's our resting potential, -70 mV. It's established because we have a separation of the ions inside and outside. It requires energy to do that. But how does the resting potential finally lead to an action potential? Now let's look at what the action potential look like. We can see that there is a huge amount of ions motion on the surface. If we zoom in, we see the sodium and potassium leak channels, but we also see a couple very crucial channels. And those are voltage-gated channels. How they work? We will start with sodium voltage-gated channel. It has a little polypeptide wall. When the voltage reaches negative 55 millivolt, it will open up. It opens so wide that allows a huge amount of sodium to rush into the cell. As we move away from that voltage, sodium voltage-gated channel is going to be inactivated, and as a result, it's going to close. There is also potassium voltage-gated channel, and it will open when the voltage becomes positive, in our case around 50 millivolts. It will be wide open and only allow the passage of the potassium, and potassium will move outside of the cell. As we look at the action potential, once we hit the threshold at negative 55 millivolts, all sodium voltage-gated channels are going to wide open. Sodium ions are rushing into the cell and therefore depolarizing the neuron. Voltage will increase drastically until it gets to 50 millivolts, and those sodium voltage-gated channels are going to be inactivated. At this point, the potassium voltage-gated channels are going to open wide. The potassium ions will spit outside and are repolarizing the neuron. It takes a while to close these channels, and because of this, we have hyperpolarization when the voltage has dropped really low. In this time, the permeability leak channels starting to work and to help to re-establish the gradient to negative 70 millivolts. And only on this stage we can have another action potential. Let us see the slow motion. We've got a quick opening of the sodium voltage-gated channel. We see a flow of sodium from outside that's followed quickly by opening of the potassium channels with outflow of the potassium. And then we can see the permeability to re-establish at the resting potential. Let us look at the action potential on the graph of the potential over time. Open up sodium, a rapid depolarization. Opening of the potassium, a polarization, and then hyperpolarization. And during this time, until we re-establish the gradient at a resting potential, we can have another action potential. Everything what we have discussed until now has happened in very short time, 2-3 milliseconds at one point in the axon. To send a message anywhere, the action potential has to move along the axon. How is it organized? Well, we have the depolarization of the segment of the axon, which triggers the depolarization of the next segment, while the repolarization of the previous segment are following, and then the polarization of the next segment and so on. Therefore, action potential happens over and over again along the axon, from the cell body to the axon terminal. When the axon terminal becomes depolarized, it splits out tiny droplets of neurotransmitter, a chemical that the dendrites of the next neuron can taste using the diffusion process. Then the entire process is repeated in this next neuron.